I should first say thank you for the organisers for inviting me to come and talk to you. I must admit the programme is quite refreshing to see so many topics in 2 days on such a wide range of vitamins, which is something you don't often get in big international meetings. I first started working on vitamins back in 1981. So I've seen quite a long history of the research developments, particularly in my own area. So what I want to do in the short amount of time I have is talk a little bit about the bioavailability and biological activity of vitamins. I then cover some more work on bioavailability of folates, which is one of my main research areas, and particularly show how our understanding of folate metabolism using human studies and improvements in analytical methodology has allowed us to get new insights into folate metabolism and bioavailability. And then just finish up with some perspectives on where the work is going in the future. Now, for most of you, this is fairly familiar. Vitamins can be classified as both water and fat-soluble. Fat-soluble vitamins are more likely to accumulate in the body, whereas the water-soluble vitamins tend to have a much faster turnover and can also be destroyed by food processing and cooking. So we need to have a constant supply of intake of vitamins for optimal health. Classification of vitamins, again, are pretty familiar. In terms of the fat-soluble vitamins, we have vitamin A, also the provitamin carotenoids and other compounds, vitamin D, E and K. And you'll hear from other speakers today and tomorrow on the sort of developments, particularly on the antioxidant front, the vitamins A and E, and also vitamin D in terms of its interaction with bone health, calcium and vitamin K. On the water-soluble front, there's a whole string of vitamins from vitamin B1 up to vitamin C. Most of the work, I guess, in terms of nutritional research has focused on the B vitamins, particularly folic acid, its interaction with vitamin B12 and vitamin B2 and B6 as well. Historically, that has sort of focused on how folic acid protects against neural tube defects and spina bifida, and the role of particularly vitamin B12 in that. If we go on and look at the bioavailability and what our current understanding of what that means, if you look back at the papers that Clive West published on bioavailability, it really summarises the sort of proportion of the nutrient in the food or the supplement when ingested, which is absorbed and then utilised by the body for normal sort of metabolic function. Now, there's a lot of factors that affect bioavailability, from the more physical parameters such as the age of the individual, the status of the gut microflora, also the food matrix and other components in the matrix that might inhibit or enhance bioavailability. And these sort of terms sort of come under the overall heading of bioaccessibility, which is really a term that describes the amount of the vitamin that's actually available for absorption. When you look at the absorption, you then come across into the more physiological factors that can affect it. And they may influence how it's distributed in the body. There may also be individual variation in the metabolism of the vitamin, particularly due to gene polymorphisms. And folate is a very good example of that. And then the entry of the vitamin into its target tissues or body fluids can also be influenced by other factors. Now, we have several methods for assessing vitamin bioavailability, and these can either be in vitro or in vivo studies, particularly either using animal or human models. Most of the human work has focused on feeding studies. And I guess in the last 10, 15 years, we've seen a lot more introduction and use of tracer stable isotope techniques for looking at vitamin bioavailability. And now again, we have the advances in the HPLC and combined HPLC mass spectrometry techniques, which now allow us to look at very, very small amounts of isotopy-labeled vitamins in blood and other tissues. If you come on to the biological activity of vitamins, and here it becomes quite complicated. And in this slide, you see the metabolic pathways of folic acid and folates. And you can see from that it's quite complex. I'll come back to the metabolic routes later. The vitamin activity really is a function of its beneficial effect in humans. The vitamin itself may require bioconversion or biotransformation from one form to another as it's absorbed and enters into the systemic circulation. And that obviously then can be influenced by different metabolic pathways. So if we come on and look at the folates as a group of compounds, as most of you know, they're essential for one-carbon transfer reactions, particularly purine and pyrimidine metabolism. Good food sources include the leafy green vegetables, liver, eggs, yeast, brown orange juice, and obviously some milk and dairy products. The complication with folate occurs in foods as polyglutamated forms, and there can be up to 9 or 10 glutamate side chains. And in order for that to be absorbed by humans, you need to cleave the polyglutamates to the monoglutamates. And then we also have folic acid, which is the synthetic form with just one glutamic acid chain. Folate monoglutamates are absorbed by essentially an active energy-dependent process, mainly in the small intestine, and that obviously can be influenced by other factors, particularly pH. So why folate research? Well, most of the historical work on folate has related to the absence of its deficiency in terms of anemia. It still can be a problem today, particularly with pregnant women, and there are various nutritional recommendations for increasing the consumption of folic acid prior to conception. And there's a great debate in terms of the levels of that, and currently they're set at 400 micrograms a day for the first 3 months of the pregnancy. In terms of nutritional deficiencies, I guess folic acid is best known for the protection of spina bifida, such as neural tube defects. It's also more recently been associated with protection against vascular diseases, particularly coronary heart disease and stroke, although its interaction there may be just a marker for something else. More recently, it's been associated with, particularly with folic acid, with potentially increasing cognitive decline in the elderly, and it may do that through interaction with vitamin B12. And there's been some publications, particularly from studies done in the United States following folic acid fortification there, where they have seen increased cognitive decline in some elderly groups. I guess the most controversial area is the role of folate, particularly folic acid, in terms of increasing or decreasing cancer risk. And again, you'll hear from other speakers today and tomorrow about the latest developments there. I think it's fair to say that currently there's no direct evidence supporting that, although there's a lot of indirect evidence, particularly from epidemiological studies and animal studies, that could explain why potentially that might be a problem. But we'll come back to that later on in the conference. So in terms of assessing bioavailability, well, there's a whole array of studies that you can use. Short-term human studies where you measure plasma responses following oral or intravenous administration of the folate. Longer-term human studies, again, particularly looking at different dietary sources. Again, using a supplement, folic acid supplement, as the test reference dose. And then we go on to the isotopic studies, where you can actually trace the folate that's administered through the body. And again, there's various protocols that you can use for looking at the absorption of your test dose. I think it's fair to say that the advances in LCMS, and particularly more recently LCMS-MS, has allowed more detailed protocols to be developed for looking at folate absorption and metabolism. Some of the work that we've been doing in Norwich has focused on developing a one compartmental model of folate absorption, which you can see in this slide, where we actually able to model and measure the absolute absorption from a test dose. One of the complications of it is that the liver actually sequesters a lot of the dose on the first pass of the folate through the body. And then you actually see a small amount being excreted into the systemic blood circulation. What you need to do is be able to measure the amount of the liver that actually takes from the dose, which is quite complicated. You also need to be able to measure the volume and distribution of that particular folate pool. So some work we've done in Norwich looking at the absorption of folates from a folic acid supplement, which you can see in this lower curve, the red triangles, versus the absorption from, again, a labelled folate from spinach, which you can see is a slightly different shaped curve. And what we found was if you feed humans a test dose either of the labelled spinach or the folate as a natural formol folate, you get a very sharp rise in absorption and then a slow decay over about 6 to 8 hours. Now, when you do the same with folic acid, you see the response is a much shallower curve, and it peaks at about 2 to 3 hours compared to about 40 minutes with the reduced folates. The slope of the excretion on both curves is quite similar, but what is different, as I said, is the actual rate of absorption. And we think this is due to how folic acid is handled in the body compared to folates. And we published a couple of papers back in 2003 and 2005 that describe some of this early stable isotope work. So we've also just completed a human intervention study looking at the absorption of folic acid as a supplement versus folate from a mixed diet. And this was a two-center randomized placebo-controlled study. And we wanted to assess whether folate status could be raised as effectively with food folates as folic acid, and then whether folate status by these methods affects cardiovascular disease risk biomarkers. And we were particularly looking at homocysteine and also pulse wave velocity, which is really a function of arterial thickness in humans. So the intervention study was a 16-week study. We had 271 volunteers from both Norwich and Sheffield, which is a much higher, has a much higher rate of coronary heart disease compared to Norwich. We did a 200 microgram dose, either from folate-rich foods or from folic acid. And in the Norwich study, we included a metformin arm, which is a 5-methyl tetrahydrofolate supplement. We did dietary assessment before and after the 16-week intervention, and then took blood samples as well before and after. In terms of the foods that we used, we portioned up folates into 200 microgram a day portions in addition to the natural amount in the diet. You can see some of the foods here in terms of orange juice, potatoes, milk and cheese, and quite a lot of brassicas and vegetables as well. The baseline data shows that the groups were randomized, and folate levels and also homocysteine levels were in normal acceptable limits compared to our national nutrient survey data for the UK. Again, I don't want to go into too much detail. There's a lot of information in this slide. This actually shows the changes in folate and homocysteine status for the 3 MTHFR genotype groups, CC, CT and TT. And then we have the 3 groups, folic acid, placebo and food. And we see quite clearly that the folic acid group increases plasma levels versus the placebo, and there was very little change in the food group on intervention. If you move to the changes in the red cell folate status, there it becomes more interesting. Again, you see a significant increase in red cell folate from the folic acid group, and there seems to be a similar increase for the 3 genotypes. If you look at the food group, although not significant, there seems to be a trend, particularly for the TT individuals, which show a small but not significant increase on intervention. And again, this trend is continued with the changes in homocysteine on intervention, where again you see a significant fall both in the folic acid group of about 3 micromolar, but we also see a fall of just under 3 micromolar in the food group for homocysteine, and that's in the TT individuals. And if you remember from the previous slide, the homocysteine levels were not particularly elevated. They were well below 10 micromolar. So there would be an indication that the baseline diet is actually producing a similar result to the supplement. So the summary results from that study, we showed that the MTHFR TT individuals had a significantly lower baseline red cell folate and a higher homocysteine compared to CC and CTs. The intervention with folic acid or food significantly reduced homocysteine in the TT individuals, and this was much greater for either the CC or the CT individuals. Elevated homocysteine in the MTHFR TT had folate and B vitamin intakes, which were certainly in excess of the current UK RNIs for those nutrients. So there was some indication that we need to maybe increase those for that particular genotype group. And that paper's just been submitted to AJCN for consideration. How do I do for time? 5 minutes, okay. And then just interestingly to talk a little bit about, we included a metformin arm in the study in Norwich. And metformin is an alternative to folic acid as a fortificant oral supplement. It's a naturally occurring 5-methyl tetrahydrofolic acid. that's now available for use by the food industry. It's also been given FDA approval in the States, where it's again possibly as a fortificant. And in this slide, we just showed the changes in increases in plasma folate from the three groups in terms of intervention at 200 micrograms. The food group showed a small increase, but not significant. The metafolin was certainly much higher than the food group, and slightly lower than the folic acid group. Now, when you take, if you are measuring the bioavailability of folate from the food group, if you took folic acid as the reference dose, you would then get an absorption of about 31%. If you did a similar thing and used metafolin as your reference dose, you would get an absorption of about 39%. So there was a significantly different bioavailability as it was using metafolin. And again, work has recently been published in the British Journal of Nutrition last month. We then went on and started to think about the differences in folate metabolism in terms of the MTHFR TT individuals. And interestingly, if you look at that particular subgroup, this was some results were taken from a paper that was published by a Dutch group in 2007. And it shows the proportion of red cell folate as non-methyl folate. Now, if you remember, most of the red cell folate in red cells is actually 5-methyl tetrahydrofolate, either as mono or polyglutamated. And you see on the left-hand side, the CC individuals generally have small amounts of non-methyl folate. If you then go to the CT individuals, you start to see some individuals that have higher proportions, up to maybe 15%, that are non-methyl folates. And then if you go to the right-hand column, you can see quite clearly that in the TT individuals, the amount of non-methyl folate in red cell is much more variable, up to something like 70% can be non-methyl folate. And we don't actually know what's causing this shifted metabolism. But we think it might be a potential nutritional biomarker for disease risk, as we know that MTHFR TT individuals seem to be associated with higher heart defects, cardiovascular disease, and potentially can have altered DNA methylation that may lead to increased cancer risk. So just quickly to finish up with some work we started last year to look at the absorption of folic acid compared to the absorption of natural folates. And this slide just shows the current thinking in terms of how folates and folic acid are metabolized in the gut. The current thinking is that folic acid is actually reduced and rearranged to tetrahydrofolate and then methylated as it goes across the brush border into the hepatic portal vein onto the liver. But we think that probably folic acid goes across as folic acid and is then handled slightly differently by the liver. And we're currently doing a study with clinical colleagues in University of Newcastle that have a group of very special patients that have a hepatic portal cannulation being inserted. Now these patients are ex-alcohol patients that are now stable, and as part of their treatment have had a cannulation put in, mainly to control the pressure of the portal vein, which can be a problem with liver disease from too much alcohol. What this enables us to do is to actually take blood samples from the hepatic portal vein, so we can actually see what type of folates are coming across from the small intestine after absorption. And we've developed a protocol where we give labelled folic acid and also labelled formyl folate, and then we can track them by taking hepatic portal brain samples and also then systemic samples, and we can then try and mathematically model the kinetics of the absorption of the different forms. And this just shows you a picture of the, it's actually called transjugular intrahepatic portal systemic shunt, or TIPS for short. And you can see this in this slide. And within the shunt, there's a very fine catheter that's inserted from which you can take the blood samples. And again, that's the picture of the shunt that you can see. So we've just completed our first subject last week. So we've collected the blood and we're now sending those for analysis. So that hopefully should start to tell us about how and what appears in the hepatic portal vein in terms of the form of the folate. So just to finish off, I think you could say that vitamin bioavailability studies have come a long way over the last 20 years, from the early animal and human studies to the types of protocol that we are now doing. I think particularly the use of human in vivo and stable isotope with modelling and advanced analytical techniques have certainly contributed to our understanding of metabolic processes in vivo. The use of more sophisticated biomarkers and particularly maybe the application of omics techniques will certainly extend our knowledge in the future. So just by acknowledgements, from my own team in Norwich, a lot of the work has been done by Tony Wright and Maria King. Jack Dainty is our mathematical modeler, and Chika is our analyst. And then Caroline and Charlotte have helped with the human studies over the years. We collaborate quite a lot with the University of Sheffield, Professor Hilary Powers and her team. We work with the University of Nottingham with the analytical techniques, particularly the OC MSMS. And then we work with our clinicians in Newcastle with Professor Dave Jones and his team. And we've had funding from a number of organisations, including Merck, and then the Food Standards Agency and BBSRC as well. So thank you very much for your attention.